Glycolysis. Glycolysis is a catabolic process where one molecule of glucose is broken down into two molecules of pyruvate, which serves as the precursors for aerobic catabolic pathways such as Krebs cycle. Glycolysis occurs in the cytosol of a cell and does not require oxygen, meaning it can function under both aerobic and anaerobic conditions. Glycolysis consists of two phases. Phase 1, known as energy investment phase, where ATP is consumed to prepare glucose for breakdown. Phase 2, known as energy payoff phase, where ATP and NADH are produced. Glycolysis consists of 10 enzyme catalyzed steps. Phase 1, energy investment phase. Step 1, glucose phosphorylation. Glucose undergoes phosphorylation and is converted into glucose 6-phosphate, catalyzed by enzyme hexokinase or glucokinase. This step requires ATP and Mg2+ ion as cofactors. When the glucose level is low, hexokinase is active, ensuring cells get energy. When the glucose level is high, glucokinase is active in the liver, leading to glycogen storage. Step 2, rearrangement. Glucose 6-phosphate undergoes isomerization, which is then converted into fructose 6-phosphate, catalyzed by enzyme phosphoglucoisomerase. This step rearranges the molecule from an aldose to a ketose. Carbonyl oxygen of glucose 6-phosphate is shifted from the C1 carbon to the C2 carbon to form fructose 6-phosphate. Step 3, second phosphorylation. Fructose 6-phosphate undergoes phosphorylation and is converted into fructose 1,6-bisphosphate, catalyzed by enzyme phosphofructokinase. This step requires ATP and Mg2+ ion. Step 3 is also known as the committed step in glycolysis. This is because once fructose 1,6-bisphosphate is formed, glycolysis must proceed. Phosphofructokinase is activated by AMP and ADP. On the other hand, it is inhibited by ATP and citrate. Step 4, cleavage of fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate splits into two triose phosphates, catalyzed by enzyme fructose bisphosphate aldolase. Fructose 1,6-bisphosphate undergoes cleavage between the C3 carbon and the C4 carbon. This is to form dihydroxyacetone phosphate DHAP and glyceraldehyde 3-phosphate G3P. Step 5, isomerization. DHAP undergoes isomerization and is converted into G3P, catalyzed by enzyme triose phosphate isomerase. By the end of this phase, each glucose molecule has been converted into two molecules of G3P. Phase 2, energy payoff phase, converts two molecules of G3P into two molecules of pyruvate. Step 6, NADH production. G3P undergoes oxidation to form 1,3-bisphosphoglycerate, catalyzed by enzyme G3P dehydrogenase. Per glucose molecule, two molecules of NAD+ are reduced to form two molecules of NADH and H+ ions. Step 7, substrate level phosphorylation. 1,3-bisphosphoglycerate donates a phosphate group to ADP, forming ATP and 3-phosphoglycerate, catalyzed by phosphoglycerate kinase. Per glucose molecule, two molecules of ATP are generated, which helps to pay off the ATP debt from step 1 and step 3. Mg2+ ion is required in step 7. Step 8, phosphoryl transfer. 3-phosphoglycerate is converted into 2-phosphoglycerate, catalyzed by enzyme phosphoglycerate mutase. Phosphoryl group of 3-phosphoglycerate is moved from the 3 carbon to the 2 carbon. Step 9, dehydration. 2-phosphoglycerate is dehydrated to form phosphoenolpyruvate, catalyzed by enzyme enolase. This reaction involves the removal of water molecules to form enol structure of PEP. Step 10, substrate level phosphorylation and formation of pyruvate. Phosphoenolpyruvate is converted into pyruvate by transferring a phosphoryl group to ADP, catalyzed by enzyme pyruvate kinase. Mg2+ and K+ ions are required for the activity of pyruvate kinase. Here's a quick recap. In phase 1, energy investment phase consumes 2 molecules of ATP. In phase 2, energy payoff phase generates 4 ATP molecules. Glycolysis produces 4 ATP but consumes 2 ATP in the investment phase, leading to a net gain of 2 ATP per glucose molecule. And here's the chemical equation of glycolysis. Glucose molecule is broken down into 2 pyruvate molecules. 2 ATP molecules are produced. 2 NAD+ molecules are used to produce 2 NADH molecules, and 2 H2O molecules are produced. In aerobic conditions, pyruvate is oxidized, and acetyl coenzyme A is formed. This acetyl group is metabolized in the Krebs cycle. Alternatively, in anaerobic conditions, pyruvate can be reduced to lactate through oxidation of NADH to NAD+, also known as the lactic acid fermentation. In yeast and some bacteria, pyruvate is converted into ethanol and carbon dioxide through alcohol fermentation, which regenerates NAD+ by oxidizing NADH. Thank you for watching. Leave like and subscribe.